structure that was initially present in the abdominal cavity. You must have learned. Then it descended outside in the scrotal wall. Right? So, the reason being that it's being placed later on outside the body cavity. The core temperature of the body is near 37 degree. So it has to maintain its temperature around or at 34 degree. Lower temperature is required. So can anyone tell few good reasons that how it's being maintained at this lower temperature? Any good reason? You must have learned Ross as well. Must know that. Common sense should be there. Okay. One of the very good reasons that the sweating perspiration that can cause the heat loss as it is not lying inside the abdominal cavity. Number two, being supplied by the testicular artery, and this testicular artery is engulfed or surrounded by pampiniform fluxes of veins. You should remember. And what happens when the artery vein they come in contact? That there is counter current heat exchange, right? So this is how the warm blood in the artery is being cooled. So the separatus gets its look of that it becomes cooler. This is the next reason to maintain its temperature at 34 degrees, right? And there is another that it has got dartus muscle, and cremaster muscle is also there, along with the movement of that spermatic cord. So the movement also helps out to lower the temperature. Why it's important? Because in females, the ovary was inside, but the gametes, they were not influenced by the heating mechanism. But these gametes or spermatozoa, they need lower temperature. That's why Allah has placed it on outer side and has given so many methods or mechanisms to keep it at 34 degrees Celsius, right? Okay. Now we have to see silently. It has got the outer covering, you can see that, which is tunica vaginalis. It has got outer parietal layer and the visceral layer, almost in the same color you can see. But a very striking color is being shown in the green. That is tunica albuginea. That is basically the capsule. And you know capsule is made up of dense irregular connective tissue. Right? So this capsule or this connective tissue becomes thickened posteriorly near the mediastinum or hilum region. Right? And you know that hilum is for the entry-exit of ducts, artery, vein, lymphatics, whatsoever. And this capsule gives septa towards the other edge, and in this way it divides it into so many testicular lobules, right? And each lobule is filled with so many folded, convoluted tubules called as seminiferous tubules or seminiferous tubules, right? Okay. Then there should be the interlobular connective tissue around them, and of course it is very well vascularized, right? So this is the look we can see at first glance. Let me show some other parts. This is the label one, we will discuss later. Okay, here you can see again the same diagram but with a different color. This commercial structure, that is the duct of epididymis. It has got upper portion, middle and lower. Lower is highlighted, which is a tail, right? Okay. This picture is showing the body of the epididymis, right, in the green color. This picture is showing the head region of the epididymis. So these three colors will tell you that which portion of this epididymis is ending up to where. Now, this is showing that where the tail of this epididymis is going to be end. It will continue as vas deferens or ductus deferens. And you know that being content along with other vessels, it has to pass through the inguinal canal through various rings and of course reaching in the pelvic cavity and we will see if any diagram comes that where it gets. Let me tell that it has to get fused with another duct to form the ejaculatory duct. We open the urethra inside traversing the prostate. Okay. After that, we'll move towards another schematic representation. So you can see the sagittal view, the tunica albuginea is there next to this tunica vaginalis. You can see the capsule and various lobules. They are being filled up with these seminiferous tubules. And a striking thing you can see that this is a blind-ended tubules. They are having on the other end a little bit straighter tubules, right? These are called as tubuli recti or vasa recta. Okay? These straightened parts of the terminal end of the seminiferous tubules, they are called as vasa recta or tubuli recti, right? And they are again entering into another network of these tubules called as Testis, right? Okay. After that, we will see they are again showing some 10 to 12 number of fine tubules called as efferent ductules or ductuli efferentes. Okay. What is going to happen? That these are entering into the head, you would say, or duct of epididymis. So it means there is a chain that has started from the seminiferous tubules and becoming the tubuli recti, then the Testis, then they are entering through the efferent ductules into the epididymis. So how we will cram them or learn? Let me show you a label diagram, and I will, okay, the same diagram with the label one. You can see that various coverings, the septas, this is the mediastinum portion, and that was the straightened part after the seminiferous tubules, right? And these are the tubuli recti. And vasa recta, they are called vasa recta, and this network is Testis. So can we see that the initial word, that if this was the seminiferous tubules, they are having the terminal straightened part, the tubuli recti, right? They are entering into this network called as Testis, right? Okay, now they have to enter into efferent ductule, which is going to join the duct of epididymic duct, you would say. Now, just remember the initial, starting first with the T, right? Later on R. Now that starts with the R, ending up with the T. TR to RT, right? To remember that which duct is first. Now after that, there is E into E. Is it easy? Now to remember that seminiferous tubules, terminal part, you would see tubuli recti, then they are going to open into the Testis. They join the efferent ductule, E into E, the duct of epididymis. This is how. Okay. Now if we cut and see the epithelium, that is the most important part of that microanatomy or histology. If we see the cut section of seminiferous tubules, they are showing the modified type of stratified cuboidal epithelium. Very important thing that can be asked in MCQ or Viva. Okay. When we cut the next tubuli recti, it will be showing the simple cuboidal. But epithelial cells, number one cell that line, that simple cuboidal is the Sertoli cell. The other are simple columnar or low columnar, you would say, or cuboidal rather, right? So simple cuboidal epithelium is there. Then in this network, Testis, again showing the simple cuboidal epithelium. They are laden with the microvilli over it and a larger single kinocilium if we see the transverse section inside. Now see this efferent ductules, when they are cut, they are showing the simple columnar epithelium. But it's being said that this simple epithelium has got few basal cell and few large taller cell, right? So giving an uneven sort of lumen of these ductules. Okay, then we are going to enter into the duct of epididymis. That would be now showing the pseudostratified columnar epithelium lined with the stereocilia, right? So, how to again recall the epithelium? The seminiferous tubules, stratified cuboidal, right? These two will be simple cuboidal, and this will be simple but now columnar, and this now pseudo-stratified columnar. So this is how we will learn that how the name of these tubules are there and which is gonna open where and which epithelium is there. Okay. The different duct pattern, and you can see the arrangement of the testicular artery and dividing plexus. Again the same schematic representation. Slide of the seminiferous tubules or testes. So what we see? First of all, if there is outer tunica vaginalis, in between you can see the cavity of it. Next, that capsular covering is tunica albuginea. We have discussed the dense irregular connective tissues there. Okay, now see that you can see various even oval rounded section of these seminiferous tubules. You saw them so many convoluted tubules. When you cut it down, you observe these lumen structure. Okay. Now this region in between this is the intertubular connective tissue or interstitial connective tissue area, right? And they are well laden with the interstitial cells. And among these cells, so many could be apart, could be macrophages, could be mast cells, could be lymphocytes. But above all is the interstitial cells of Leydig. As we know that testes, again, an important organ being exocrine and endocrine, right? So it has to do the function of gametogenesis and steroidogenesis. So gamete formation is around this seminiferous tubules, right? So exocrinal function, we can call it a simple compound gland, if we say it is exocrine. And here, outside them, this interstitial area, that shows the interstitial cells of Leydig. They are supposed to produce the testosterone. Means hormones are produced over there, right? So this endocrine function is being performed by them. Okay. Now we have to discuss this cell series, and what epithelium we saw here? The modified stratified cuboidal, the rare parts, right? ओके अब हम इसको देखते हैं, थोड़ा सा बड़ा इसका इंप्रेशन, एक आपको कट सेक्शन दिखाया जा रहा है. ओके, दैट वाज द रिम. इफ यू कैन ऑब्जर्व, इफ आई कंप्लीट इट, बिकॉज अ लिटिल पोर्शन हैज बीन शोन हियर बट नॉट हियर. ओके, it's being lying on its outer side, the myoid cell. The outer margin of each convoluted tubule has got these, and they are supposed to give contractility, right? Okay. Then we have got, as we mentioned, stratified cuboidal. So there are different cell series. First cell present along this basal layer, you would say, is the spermatogonia. Right? These are the spermatogonia present on the as a first series, and these are the immature most cell found even in the kids. Right? Okay. Then they have to become the primary spermatocyte. Right? This primary spermatocyte is the largest among all, and it will finally change into or mitotically divide and will become the secondary spermatocyte, right? Okay. Then it has to again form the spermatids. These spermatids now get mature, transform, not now mitotically divide, transform into spermatozoa, has spot sperm. This is the lumen, right? Okay. Intervening, you see the fertilizer. This is a histological section in which you can see them as label diagram, but for understanding I think I have to make it. Please have a closer look of it, and you can take a pic of it. Let me make the, this is the seminiferous tubules, there could be the other one, and this is the area where interstitial cells of Leydig are present. which are steroid producing cells, well laden with the mitochondria, tubular crystal could be there, and all the organelles you mentioned, smooth endoplasmic reticulum, Golgi apparatus, ribosomes, so and so. Right? They are filled with the lipid, and they are supposed to produce what? Testosterone. Right? And these Leydig cells, they do not have the secretory granules. Why? Because as soon as they produce the hormone, they release it into the nearby blood capillaries of blood vessels. So they don't need to be store their secretion. Right? As soon as they produce, they release it. Is it clear about these cells? Okay. अब हमने डिस्कस करना है जी ये stratified cuboidal epithelial series. बाहर तो हमने इसके डिस्कस कर लिया कि that was the myoid cell to give contractility from all sides. Okay. If I make the first cell series, the spermatogonium, they are of two basic types. Spermatogonia type A and type B. Now again that type A is in the form of a dark type A and pale type A. Okay, what this dark type A spermatogonia appears, it has got a darker stained nucleus, and the nucleolus is located on one side. It is acting as a dormant, in a dormant phase, but acts as a stem cell. It rarely undergoes I would say, mitotic activity, but it has to maintain its own number and the pale type. ये जो डार्क सेल थे, ये अपना नंबर मेंटेन करते हैं और पेल का नंबर मेंटेन करते हैं. इसलिए ओके. अब ये जो पेल टाइप है, इनका क्या काम है? इन्होंने अपना नंबर मेंटेन करना है एंड विल माइटोटिकली डिवाइड टू फॉर्म द बी टाइप. इज इट क्लियर? तो आप यहाँ पे इसको बना सकते हो कि आपने बी टाइप भी बनाना है, आपने ये इनको ए टाइप मेंशन करना है, एक को आप पेल कर लेंगे, एक को आप डार्क कर लेंगे, डिपेंडिंग अपॉन द पेरेंट्स ऑफ द न्यूक्लियस, हाउ इट इज प्रेजेंट, हाउ इट इज शोइंग द वेसिकुलर न्यूक्लियस, finally dispersed chromatin could be part. Okay, it has to now mitotically divide and become the largest cell series called as primary spermatocytes. Right? After that, it has to now divide again to form the secondary spermatocytes. Right? Now, what they are going to do? Now they are supposed to form the spermatids, and finally they transform, undergo spermiogenesis. Now spermatogenesis was the initial process in which mitotic activity was taken place. Then here the primary spermatocyte, that is the place where the premeiotic now division has to occur. And what is going to happen? That these cell series, the Gonia series, they have got 46 number of chromosomes, isn't it? But when this primary spermatocytes, they again have 46. But when they form this secondary type, their chromosomes are now, after meiotic division, they become 23, isn't it? Now this number will be maintained in the rest of the series. Is it clear?
Yes.
Okay. Another important thing that there is, if I complete it, there are so many cells here and here, but in between they have got a cell called as satellite cell. That is a tall, heighted cell that is of columnar variety, starting from the basement membrane up to the lumen. It has got a nucleus that has got folded nuclear envelope, one of its important features, right? It itself is of paler color, but the nucleolus is very darker, right? So that is a sartolite cell. So what is going to happen? That this sartolite cell laterally has got processes, and from the other side there is another sartolite cell that is present, and they are forming the junctional complexes, gap junctions, occluding junction, so and so. And this is the important thing. These satellite cells They do not divide mitotically. They are present here. Let me make other cell series. The larger series remains here. Another sartolized cell comes. Gonia was there. Okay. What they do? They, after forming that junctional complex, produce the blood-testis barrier. The most important thing, the question can come that how blood-testis barrier is formed, which cell is responsible, and how do you understand that, how do you explain it? Okay, this blood-testis barrier, it has to do what? It has to do a lot many function, or sartolized cell has produced a thing that is dividing this whole area into two compartments. This is the basal compartment and this becomes the adluminal compartment. Adding towards lumen. Is it clear? इस तरफ से अगर मैं बनाऊं, this is a sartolized cell, okay. इस तरफ से भी sartolized cell आ रहा है. ये इन्होंने barrier बना दिया. अब ये वाली जगह, this is the basal compartment. ये lumen की तरफ वाला adlumen compartment. Very important. अब क्या होता है? इसके चारों तरफ, as we have told कि ये testigious cells हैं और ये well vascularized area, even it is called as tunica vasculosa, again like that of which is present in the myocardium, well vascularized. Now these blood vessels and blood cells have to interact with this cell series, the Gonia series, the primary spermatocytes. लेकिन होता क्या है? They know कि इनका नंबर भी 46 है और ये इसको पहचानते हैं, they don't harm them. Is it clear? This basal compartment has got cell series which is recognized by the blood cells and they leave them, yeah, we know you. But if there is no such blood barrier, the series which has got 23 number of chromosomes, the secondary spermatocytes, the spermatids and gonia, they could have taken as a foreign body and the blood cell could have killed them. So Allah has made your body accordingly that there is a well mechanism called as blood-testis barrier. Okay. Next question arises that this cell series is being nourished through these blood vessels. So from where they get their nourishment? Question can come in your mind. So can you guess from where they can get their nourishment now? Yeah, sartolized cell is gonna nourish them because it also is giving support to all, giving nourishment to this series and saving these cells from the attack of the blood cells, right? Because they have got different surface marker. Is it clear? Okay. Now another question, if there is a complete occluding junction, how the cell series that passes here? It seems impossible, ना? Get बंद है, तो कैसे ये सीरीज जो 46 थी meiotically divide होके 23 हुई, तो ये कैसे यहाँ पे पहुँच गई? They could be. So Allah has again made a mechanism that there is a transient opening of the gate, right? So in this way, the cells is still, these germ cells still make connection with it and slip down, get here, and the gate closes. ये बिल्कुल ऐसी है जैसे अगर डोर को हम बंद कर दें, कोई फॉरेन बॉडी, कोई एनिमी, कोई डिफरेंट बंदा अंदर नहीं आएगा. बट वी नो कि स्टूडेंट इज कमिंग, तो हम उसको हल्का सा बोलते हैं, he or she is allowed to come in and we close it. So this fine mechanism is running without the awareness of a person. Allah is saving so many cells from the attack of the blood cells. So very important. Most of the time the note comes that kindly discuss the blood testis barrier or how this progeny or 23 number chromosomes containing cells are being saved. So you have to tell all these things, or the question can be asked that from where they get their nourishment. So in any way you have to explain this whole mechanism. So this sartolized cell giving support, nutrition, and it also produces some other thing like Müllerian inhibitory substance, right? To regress the paramesomorphic duct system, that system that is a part of female reproductive system. Another important thing that it has to produce androgen binding proteins because we need more testosterone here, right? The third thing is the production of inhibin, right? Inhibin is doing what? फीडबैक मैकेनिज्म जाएगा टुवर्ड्स द पिट्यूटरी दैट द एफएसएच, जिसके अंडर कंट्रोल आप देख रहे थे सारा आपको जो भी गोनाडोट्रोपिक हार्मोन्स होते हैं एलएच एफएसएच, तो एफएसएच को एक्सेसिव जो अमाउंट होगी इसको इनहिबिट करेगा. ठीक है, सो दिस इज हाउ सो मेनी फंक्शंस आर बीइंग परफॉर्म्ड बाय these cells. Okay, so after that we have to discuss various other pictures. Let me explain it. Is it clear so far?
Yes.
Okay. Gonia was first, then primary spermatocytes was second, then secondary spermatocyte was the third, then spermatids were the fourth, then spermatozoa. ऐसे ये कुछ ये बेस से ल्यूमन की तरफ जा रहे हैं. So make the initial that you should write go. Please see tids थे आप ये इसका T skip कर लें, ले लीजिए, the Z को collect कर लें and you can write the zoo. So read it, go please see the zoo. Now you will never forget कि आपने spermatogonia most immature basal region पे लगाना है, फिर आपने प्राइमरी स्पर्मेटोसाइट एंटर करना है, फिर आपने सेकेंडरी स्पर्मेटोसाइट लिखना है, फिर आपने स्पर्मेटिड्स मेंशन करना है, देन जोआ. अगर आपने इसको रिवर्स कर लिया, जोआ इधर फिट कर दिया है, प्राइमरी इधर कर दिया, तो आई थिंक ना तो आपका वाइवा बेहतर कंडक्ट होगा और ना ही आप इसको रिटर्न ठीक तरीके से लिख पाओगे. इम्पोर्टेंट टू राइट कि इनका आपने कैसे सेल सीरीज लिखनी है, और अमंग ऑल द लार्जेस्ट वन इज द प्राइमरी वन, राइट. और आपको ये क्लियर होना चाहिए कि बेसल कंपार्टमेंट में कौन सी प्रोजेनी रहती है, कौन से सेल सीरीज रहते हैं, और ये एड्यूमिनल कंपार्टमेंट में कौन से होंगे. दैट वाज रिलेटेड, सो आई हैव टू टेल यूज कट सेक्शंस. The myoid cells in the peripheral region is being shown, and this hole is labeled as germinal epithelium. Again can be asked, what is germinal epithelium that lines the testes, and what is the germinal epithelium that lines the ovaries? That was simple cuboidal, it is stratified cuboidal, right? And interstitial cells of blading are present in between. That we will discuss later on, or we have done. Now seminiferous tubules, the series is being written. The Sertoli cell, one Sertoli cell, okay, Sertoli cell margins are not very clear because they are making junctional complexes with the other. Just the nucleolus and the nucleus is being shown. But if I draw it, it is the taller cell. And rest of the series is the gonia, the primary spermatocytes, you can add the secondary, then spermatids and spermatozoa. And intervening area will be showing the blading cells, right? And on the margins, you observe the smooth muscles to give the contractility, the myoid cells. Okay? That was just that they are convoluted, how much long, what is the diameter, being surrounded by the myoid series cells. And myoid cells are what? Contractile in nature and help propel the spermatozoa towards the tubules. Again, cell series individually made to make you remember that the basement membrane, the gonia, the primary, the secondary, the spermatids, and the zoa. This is how. And they are in this form that the head is down and the tail region is towards the lumen. Okay. Again, a picture showing you the tubules, the interstitial tissue, and the cut sections. Again, a clear picture highlighted the where lies the septa, where lies the albuginia, and the straightened tubules or tubuli recti or vasa recta. Then you see the reti testes in the mediastinal region. Then you see the efferent dictule and the epididymis, right? So this is the way you can now, from here you can observe the tunica vaginalis and albuginia, which is the capsule. This was the repeat thing. So again, showing various ducts, the blood vessels, and the lobular portion. So that was a single trunk is taken, the Sertoli cell, the cell series. So either you start from base or draw lumen from this side, or you draw from the other side. One and the same thing. आपने इसको अपसाइड डाउन नहीं करना है किसी तरीके, ये याद रखना है कि ल्यूमन की तरफ कौन सी सीरीज है और बेस की तरफ कौन सी सीरीज है, इसलिए सो मेनी पिक्चर्स आर टेकन जस्ट टू मेक यू रिमेंबर. Okay, this is taken from Janquera. Very important because the number of chromosome is being written to make you clear. There is the spermatogonia with the 46 number. The same number is maintained in the primary spermatocyte, and then you get the tight junctional complex in between the one centralized cell and the other. Then comes the secondary spermatocyte, and here the abdomen compartment has started. Number is now into 23. Okay. Then they are changed into spermatids. Okay. Now the spermatozoa, and the 4 number is maintained after the single cell series. Okay. Okay. After that, you can see the next portion. Let's start with here that it has got a head, then a constricted portion which is called the neck, then the tail portion which is showing the middle piece. The longer one is the principal piece, and the terminal is the end piece, right? So the neck region will be showing the centriole in it, right? And then rest of them, they have got axoneme, which could be in the form of doublet being surrounded by the 9 pair of, again, doublet microtubules. That is how the axoneme is being formed, and the mitochondria is there to give energy. And finally, we will see the end piece that is showing the terminal portion of the tail region. Again, this is another slide of the ductus epididymis. So that was just to make you clear the labeling of different. These are the tubular recti और vasa recta और straightened tubules. इनके 3 नाम दिए जाएंगे. तीनों एक ही चीज है, इसको आप एक दफा समझ लेंगे then it will be clear. और उसके बाद ये रेटी टेस्टिस है, ये TR और RT जरा आपके लिए ज्यादा आसान हो जाएगा. इसके बाद ये E-फ्रंट E से स्टार्ट हो रहा है और ये एपिडिडमस है. I think that was all for today. Again, this is taken from Like Hussain, the schematic view showing various ducts, the coverings, so and so. Again, the same picture to show you things. Thank you very much. Now, the capsule, the tunica albuginea. Then you see so many rounded cut section of seminiferous tubules, right? ये मेरे ख्याल आप में से कोई आके डिस्कस करे. कौन आएगा आज? Any new face? Anyone? आ जाए. अभी आपने सारा पढ़ा है, जस्ट आपने से बताना है. आप पास भी हो रहे हैं. Sort of. डॉक्टर साहब आप आ जाए फर्स्ट. जी आप आ जाइए. It's not that difficult.
सबसे पहले उसकी आउटर बाउंड्री है जिससे which is made up of myelite cells which are contractile in nature.
अच्छा, हम इसको माइलॉइड जो आपने बताया तो सेमिनिफरस ट्यूब्यूल्स के इर्द गिर्द होंगे. इसको आप स्टार्ट कर लीजिए ट्यूनिका एल्बुजिनिया से. ठीक है. कैप्सुलर कवरिंग.
Capsular covering test irregular connective tissue is tunica albuginea.
ठीक है.
उसके नीचे जो है myelite.
माइलॉइड में आपको ये जितने कट सेक्शन दिख रहे हैं ये क्या शो कर रहे हैं? सेमिनिफरस ट्यूब्यूल. और इनके दरमियान आपको गैप मिल रहा है इंटरस्टिशियल. यहाँ पे क्या पाया जाता है?
यहाँ पे लेडिंग सेल्स.
लेडिंग सेल्स. अच्छा, आप इसको थोड़ा सा रोटेट कर सकते हैं. लेट मी हेल्प यू. सो दिस इज अ सेक्शन ऑन 4x, राइट? सो लेट मी टेक इट. ये जो स्ट्रेटिफाइड क्यूबॉइडल एपिथेलियम थी, अगेन कम हियर. जी, अब इसको बताइए कि अगर हमने ये सेल सीरीज बतानी है कि स्पर्मेटोगोनिया कहाँ पर है और सो एंड सो, तो आप कहाँ से स्टार्ट करेंगे?
सबसे पहले एपिकल साइड से स्टार्ट करेंगे. तो एपिकल साइड से सबसे पहले माइलॉइड सेल्स
बेसल रीजन से. बेसमेंट मेम्ब्रेन पे पड़ा होता है. ठीक है.
पड़ा होता है. उसके बाद स्पर्मेटोगोनिया
गोनिया
सीरीज,
कवर्स द फर्स्ट इमेच्योर सीरीज.
फर्स्ट इमेच्योर सीरीज. उसके बाद प्राइमरी स्पर्मेटोसाइट्स, द लार्जेस्ट वंस.
ओके.
उसके बाद सेकेंडरी स्पर्मेटोसाइट्स,
ठीक है.
स्पर्मेटिड्स
स्पर्मेटिड्स आएगा.
एंड देन मैच्योर स्पर्मेटोजोआ.
वेरी गुड, शाबाश. थैंक यू. बैठिए. और ये दरमियान में ये जो आपको इंटरस्टिशियस एरिया है, इसको फोकस करें तो हम यहाँ कह सकते हैं कि इंटरस्टिशियल सेल्स ऑफ ब्लीडिंग है. इसका मतलब है ये फैक्ट्री गैमेटोजेनेसिस का काम कर रही है और यहाँ पे आपको हार्मोन्स की प्रोडक्शन दिखाई दे रही है. तो इधर 10x पे फोकस किया जाए so you are able to tell that these are the cut section of seminiferous tubules or the slide of testes आपने इसको लिखना है because we are asking you the name of organ, not the individual tubules inside. And the cell series is lying over here. So this is how you are supposed to observe the slide. So that was all for today. Thank you.